Sometimes there are genes that we want to suppress. This technique has been used successfully to identify gene function. One technique used for gene silencing, or knockdown, is RNA interference, also known as RNAi. The initial step of RNAi involves the introduction of double-stranded RNA into C. elegans. This can be done using E. coli. C. elegans ingesting E. coli with double-stranded RNA present provides the means for this introduction. Somehow our C. elegans transformed from a hermaphrodite into a male. We're not sure how that happened. Now, taking a closer look inside C. elegans, we can see the black segment which represents the sequence of double-stranded RNA introduced by the ingestion of E. coli. Problematically, this sequence of double-stranded RNA is too long. In order to function in an interference capacity, it must be chopped into smaller sequences that are around 20 to 25 base pairs in length. This chopping is accomplished via the dicer protein, which is represented by the yellow Pac-Man cutout. This splices the double-stranded RNA into shorter 20 or 25 base pair segments, which is represented now by the red sequence of double-stranded RNA. Now, the RNA-induced silencing complex, also known as the RISC complex, binds to the short segment of double-stranded RNA. The argonaute protein, represented by the yellow badge component of our RISC complex, facilitates the splitting of the short sequence of double-stranded RNA into two single-stranded segments of RNA. One remains bound to the RISC complex, and the other will be degraded in the cytoplasm. The single-stranded segment of RNA, still bound to the argonaute protein, will move towards a sequence of mRNA. It looks for its corresponding base pairs. Once it's found these base pairs, argonaute will undergo a conformational change that results in the splicing of mRNA. The corresponding base pairs that match the argonaute-bound single-stranded RNA will be removed from the mRNA. The removal of this segment means that it's no longer a part of the mRNA sequence and will no longer be translated by ribosomes. Thus, the mRNA that represents gene expression is silenced, no proteins are produced. In this manner, genes can be silenced without having to alter the genome itself. And that is RNAi.